The components of nucleic acids. DNA is found primarily in the nuclei of living cells. Meanwhile, RNA is found in the nucleus and mitochondria, but it's mainly located in the cytoplasm. Both DNA and RNA are biopolymers consisting of long linear molecules. The repeating structural units, or the monomers, of DNA and RNA are called nucleotides. Nucleotides, in turn, are composed of three simpler components: a nitrogenous base, a sugar, and a phosphate. So, a nucleotide is the repeating structural unit or the monomer that we find in the molecules of DNA and RNA, which are polymeric. So, what is DNA and RNA composed of? Well, they're composed of nucleotides. And a nucleotide is going to contain a nitrogenous base, and there's two classes, pyrimidines or pyridines. Then it's going to have a sugar, and there's two types of sugars. We can either have ribose or deoxyribose, and then they both are going to contain phosphates. Each of the five bases commonly found in nucleic acids are heterocyclic compounds that can be classified as either a pyrimidine or a purine, the parent compounds from which the bases are derived. Purines, one of the classes of nitrogenous bases that make up nucleic acids, are broken down in the body into uric acid. When too much uric acid accumulates within joints, a condition known as gout can arise. The three pyrimidine bases are uracil, which we abbreviate with the letter U, thymine, for which we use the letter T, and cytosine, for which we use the letter C. Whereas adenine and guanine, A and G respectively, are the two purine bases. Adenine, guanine, and cytosine are found in both DNA and RNA, but uracil is ordinarily found only in RNA, and thymine only in DNA. Structural formulas of the five bases are shown here, and you should try to familiarize yourself with these. All right, let's take a look at example 13.1. Indicate whether each of the following bases is a purine or a pyrimidine. Also state whether it is found in DNA, RNA, or both. And you can see here that cytosine is a pyrimidine. I have the structure of pyrimidine drawn up here in the top in blue. And so we'll put here that cytosine is a pyrimidine. And if you remember, cytosine is found in both DNA and RNA. All right, next we have uracil. And again, you can see that uracil is a pyrimidine. Pyrimidine. And it is only found in RNA. So only in RNA. And last we have guanine. And you can see that guanine is a purine. And guanine is found in both DNA and RNA. The sugar component of RNA is D-ribose, as the name ribonucleic acid implies. In deoxyribonucleic acid, which is found in DNA, the sugar is deoxyribose, which lacks a hydroxy group at the second position of the heterocyclic ring. The carbon atoms in the sugar are designated with the number followed by a prime to distinguish them from the atoms in the bases. Both sugars are present in the beta configuration in nucleotides. And you remember that these are called beta because the hydroxyl group at carbon 1 is pointing up. Now, phosphate, which is the third component of nucleotides, is derived from phosphoric acid, which has the formula H3PO4, which under cellular pH conditions exists in its ionic form. So if I draw the Lewis structure of phosphoric acid, it looks like this. Okay, so we have three OH groups, and so at physiological pH, you're going to lose two of the hydrogens, and you end up with negative charges here. And so that's our phosphate. The adenosine nucleotide shown in the equation that's on this slide is found in RNA. And the reason we know that is because the sugar is ribose instead of deoxyribose. The adenosine nucleotide shown in this equation is found in RNA, and you can see that it's in RNA because the sugar is ribose, not deoxyribose. Now, this equation shows that the base in the nucleotide is always attached to carbon 1 of the sugar, and that the phosphate is generally located at the 5 position. And of course, these are 1' and 5' respectively. And thus, the general structure of a nucleotide can be shown as is represented here, and it can be simplified even more to this here, where we have our sugar, then we have our base coming off of it, again at carbon 1', and we have our phosphate coming off at carbon 5'.